Number 1. The Spiny-Headed Worm. Start with an animal that shouldn't work. It has no mouth, it has no throat, no stomach, no intestine, and no anus at any point in its life. The phylum is called Acanthocephala, from the Greek for thorn and head, and there are roughly 1,200 described species. None of them eat in any sense you would recognize. Nutrition is absorbed straight through the body wall, a living sheet called a syncytial tegument, threaded with a canal system that circulates fluid where a gut should be. The animal is a tube of absorption with a weapon on the front. That weapon is the reason it belongs here. The front end carries a proboscis studded with rings of curved hooks. It is not pushed out by muscle in the way you might imagine. It is everted hydraulically, driven outward by fluid pressure from a muscular sac called the receptacle, so the hooks turn inside out into the tissue and lock. Once anchored, the worm does not let go. Removal tears. In the ocean, these worms belong to the largest animals alive. Bulbosoma matures in baleen and toothed whales. Corynosoma matures in seals and sea lions. The cycle begins far smaller, in krill and amphipods, where the larva develops into a stage called a cystacanth, visible as a bright orange body inside the crustacean. Mackerel, cod, herring, salmon, and squid then eat the crustaceans and carry the larvae in their flesh without changing them at all. They are storage. Nothing develops, nothing dies, the larvae simply accumulate. Then a person eats the fish raw, and the worm anchors into a wall that was never meant for it. Japanese case reports going back to the 1970s describe sudden abdominal emergencies traced to raw whitebait, mackerel, and squid. The damage is mechanical rather than chemical. Tissue dies around the hooks, a granuloma forms, and in the worst cases, the bowel perforates and the abdomen fills. Several patients have only been diagnosed on the operating table. A case in Alaska reported in 1983 recovered Corynosoma villosum from a patient who had eaten raw salmon. The early clue is almost always the same, an eosinophil count climbing for no obvious reason. Number 2. Capillaria Philippinensis. In 1963, a man from Ilocos Norte in the northern Philippines died of a wasting illness nobody could name. He had lost enormous amounts of weight. His abdomen was swollen with fluid. At autopsy, his intestine was found to contain thousands of worms so fine they were barely thicker than a human hair. Over the following years, the villages of Pudok West and the surrounding coast produced more than 1,000 infections and dozens of deaths. Untreated, the case fatality rate in those first outbreaks was reported as high as 30%. The worm was described formally in 1968 by Chitwood, Velasquez, and Salazar. The adult is tiny, 2 to 4 millimeters long, and on its own it should be trivial. What made it lethal is a trick almost no other intestinal worm can perform. Most helminths cannot multiply inside their final host. Eggs must leave the body, find an intermediate host, and come back. Capillaria Philippinensis broke that rule. Some of its females do not lay eggs at all. They release live larvae directly into the intestine, and those larvae mature there, and then they reproduce there. The infection amplifies itself with no outside step. A person who swallowed a handful of larvae in a single meal can carry more than 200,000 worms weeks later. The natural hosts are fish-eating birds, which seed brackish lagoons and estuaries with eggs. Small fish in the genera Hypsiliotres and Ambasses pick them up. And the fish are eaten whole and raw, often with alcohol, as a local custom. What follows in the patient is chronic watery diarrhea and a loud, constantly moving gut. The intestinal lining is stripped until it can no longer hold protein, and albumin and electrolytes leak away. The body swells while the muscle disappears. Death, when it comes, usually arrives through heart failure or an electrolyte collapse rather than the worms themselves. Diagnosis depends on finding eggs shaped like peanuts with flattened plugs at each end, and because egg output rises and falls, a single negative stool sample means nothing. Treatment is albendazole for 20 days. Shorter courses fail because the autoinfective cycle simply restarts. Cases have since been confirmed in Thailand, Japan, Taiwan, Korea, Egypt, Iran, and Colombia. Number 3. Gnathostoma Spinigerum. In 1836, a young tiger died at the Zoological Society of London. Richard Owen, who would later coin the word dinosaur, opened the stomach and found a tumor-like nodule with a worm inside it. He described it, and the animal has carried his description ever since. In its proper hosts, cats and dogs and wild cats, Gnathostoma is well behaved. The adult settles in the stomach wall and stays there. The head bulb carries 4 rings of hooklets, and the front of the body is covered in rows of backward-facing spines. But in the right animal, those tools are used once, and then the worm stops moving. In a human being, it never stops. The larva that arrives from raw fish cannot mature, cannot find the stomach wall it is looking for, and so it keeps searching. What the patient sees is a swelling that rises somewhere on the body, lasts about a week, and then disappears, only to reappear somewhere else entirely. The worm is moving under the skin. Documented cases have continued that way for 10 to 12 years in a single person. The route into people is the usual one. Copepods of the genus Cyclops carry the first stage. Fish, frogs, snakes, eels, and birds carry the second. Snakehead fish, swamp eels, and catfish are the classic sources across Southeast Asia, and Thailand and Japan were for a long time the world's centers of the disease. Then it arrived in Latin America. From the 1970s onward, Mexican states including Sinaloa, Nayarit, Guerrero, and Veracruz produced a steady epidemic driven by ceviche, and molecular work later confirmed the agent as a separate species, Gnathostoma binucleatum. The danger is not the swelling, it is where the worm goes when it stops traveling outward and starts traveling inward. It reaches the lung, the bladder, the ear, and the eye. When it enters the spinal cord or the brain, it produces sudden, severe, radiating nerve pain, and then weakness, and then paralysis. Spinal fluid comes back stained with old blood and packed with eosinophils. Mortality in central nervous system cases has been reported between 8 and 25%. When the worm surfaces near the skin, surgeons cut immediately, because that is the one moment it can be finished for good. Number 4. Heterophyes Heterophyes. Theodore Bilharz found this one in a Cairo autopsy in 1851. He is remembered for schistosomiasis, which still carries his name in older texts, and this was a smaller discovery in every sense. The adult fluke is between 1 and 1.7 millimeters long. It is among the smallest flukes that infect human beings, and you could lose several of them on a fingertip. It attaches with 2 suckers and a third structure that most flukes do not have, an armed genital sucker called a gonadal, which grips the intestinal lining like a clamp. The cycle runs through brackish water snails, Pyranella conica in Egypt and Cerathidae species across Asia, and then into estuarine fish, particularly gray mullet and tilapia. In Egypt, the vehicle is famous. Fasik is salted, fermented gray mullet, eaten at Sham el Nessim, the spring festival that predates both Christianity and Islam in the country. Salting is supposed to make it safe. It frequently does not. The encysted larvae in the flesh survive the cure, and every year the dish produces cases. The intestinal disease is unremarkable. Colicky pain, mucus, chronic loose stools, sometimes nothing at all. What makes this fluke dangerous is not the animal, but its eggs. They are extremely small, around 30 micrometers by 17, small enough to slip through the damaged intestinal lining and enter the lymphatic vessels and the small veins. Once they are in the circulation, they travel. Where they lodge decides the outcome. Eggs driven into the heart muscle produce focal inflammation in the myocardium, and heterophyed egg embolization has been implicated in fatal cases of heart block in the Philippines, described in the 1960s. Eggs reaching the brain and spinal cord produce seizures and focal deficits. Praziquantel will clear every adult worm in the intestine within a day, and it will do absolutely nothing about the eggs already sitting in cardiac tissue. That damage is permanent. There is one more trap. Under a microscope, heterophyed eggs look very much like the eggs of the Chinese liver fluke. The 2 parasites need the same drug, so the mistake is often invisible, but they carry entirely different long-term risks, and one of those risks is cancer. Number 5. Spirometra. There is a paper published in Genome Biology in 2014 by Bennett Berryman and colleagues that reads more like a case of surveillance than of medicine. A patient in Britain had a lesion in the brain. It was imaged and then imaged again, and the lesion had moved. Over 4 years of scans, it traveled roughly 5 centimeters across the brain tissue. When it was finally removed, it was a worm, and the team sequenced its genome directly from the specimen. The result came in around 1.3 gigabases, roughly 10 times the size of the related Echinococcus genome. The animal is called a Spargonum, It is the larval stage of a tapeworm in the genus Spirometra, whose adults live in the intestines of cats and dogs. In humans, it never becomes an adult at all. It stays a larva, a flat ivory ribbon anywhere from a few centimeters to more than 30, and it moves. There are 3 ways in, and the third is the one people do not expect. The first is drinking untreated water containing infected copepods. The second is eating undercooked frog or snake. The third is applying raw split amphibian flesh directly to a wound or to the eye, a traditional poultice practice documented across parts of Southeast Asia. That route produces ocular sparganosis, in which the larva enters the orbit and the eye swells and vision goes. Under the skin, the larva carves a channel and pushes forward with a blunt, wrinkled head. On magnetic resonance imaging, the track it leaves is distinctive enough to have a name, the tunnel sign, a winding, enhancing line marking where the animal has been. But the diagnostic feature that settles it is simpler than any sign. The lesion is in a different place on the next scan. Surgery is the answer. Praziquantel, which handles most tapeworm problems, penetrates the spargonum poorly and cannot be relied on. Cutting the larva out intact is the definitive cure, which is why extraction is attempted even from the brain. China has documented over 1,000 infections, and there is a rarer form still, proliferative sparganosis, where the larva branches and buds and spreads through the body. Fewer than 20 confirmed cases exist worldwide. Almost all of them were fatal. Number 6. Paragonimus Westermani. Konrad Westermann was a zoo curator in Amsterdam, and in 1877 he found a fluke in a Bengal tiger that had died in his collection. Within a decade, human cases were being described in Taiwan and Japan, with Ringer and Manson among the early observers. More than 50 species are now recognized, of which around 10 infect people, and North America has its own Paragonimus kellicotti, which produces cases along the Missouri and Mississippi river systems from raw crayfish eaten on canoe trips. The adult is a plump reddish-brown animal shaped like a coffee bean, 7 to 16 millimeters long. It needs a freshwater snail first and a freshwater crab or crayfish second, and it reaches humans through raw crab dishes, including the drunken crab preparations of East Asia in which live crab is marinated in alcohol rather than cooked. What the fluke does next is the reason it is on this list. It exists in the duodenum, then it does not stay in the intestine at all. It bores through the intestinal wall, crosses the peritoneal cavity, tunnels through the diaphragm, enters the chest, and settles inside the lung. There it finds a partner, and the pair build a fibrous cyst around themselves in the lung tissue. The cyst opens into a bronchiole, which is how the eggs escape. The patient coughs them up. That is where the danger becomes practical. A patient presents with a chronic cough and rust-brown, blood-streaked sputum. The chest radiograph shows ring shadows, patchy infiltrates, and fluid around the lung. In every endemic region on Earth, that combination reads as pulmonary tuberculosis. Patients have been placed on anti-tuberculous drugs for months with no improvement, while the actual cause sat in the sputum sample the entire time. The worst version happens when the flukes miss the lung. Ectopic paragonimiasis puts the animal in the wrong place, and the worst wrong place is the brain. Cerebral paragonimiasis produces seizures and visual field loss, and leaves behind calcifications with a distinctive soap bubble appearance on imaging. Before control programs, cerebral cases made up a meaningful share of neurological admissions in Korea and China. Praziquantel clears it in 2 to 3 days. Recognizing it takes considerably longer. Number 7. Angiostrongylus Cantonensis. Chen described this worm in 1935 from rats in Guangzhou. 9 years later, in Taiwan, Nomura and Lin Nura connected it to a human patient with meningitis. The adults live in the pulmonary arteries of rats, which is where the common name comes from. And the larvae are coughed up, swallowed, and passed out in rat droppings, where snails and slugs eat them. One snail did more than any other to make this a global problem. The giant African land snail, Lysachatini fulica, was moved around the world deliberately, as food, as a curiosity, and as an accident of horticulture. It carries the larvae efficiently: freshwater prawns, land crabs, coconut crabs, frogs, and monitor lizards carry them too. And there is a route that requires eating no animal at all, because a slug crossing a lettuce leaf leaves larvae behind in the slime. In a rat, the larva completes a tidy circuit through the nervous system and returns to the lungs. In a human, it enters the central nervous system and cannot get out. What it causes is eosinophilic meningitis, defined by more than 10% eosinophils in the spinal fluid. The patient has a headache described in the literature as among the most severe encountered in clinical practice, along with neck stiffness, vomiting, and strange crawling or burning sensations across the skin. Incubation is usually 1 to 3 weeks. In the ocular form, the worm can sometimes be seen moving in the front chamber of the eye. Treatment carries an unusual problem. Killing the worm makes the inflammation worse, because a dead larva in brain tissue provokes a stronger immune response than a living one. Anthelmintics are therefore given with corticosteroid cover, and a randomized trial in Thailand by Chopman-Cole and colleagues established that a 2-week course of prednisolone shortens the headache. The parasite is now spread to Australia, Hawaii, the Caribbean, Brazil, and the southern United States, carried by rats and mollusks in ballast, cargo, and nursery plants. Hawaii records the largest American cluster, and there is one case almost everyone has heard without knowing its name. In Sydney, in 2010, a young man ate a slug on a dare. He was severely disabled for 8 years, and he died in 2018. Number 8. Cymothoa Exigua. This one is a crustacean. It belongs to the isopods, the same order as woodlice and pillbugs, and it looks like one, pale gray and segmented with black eyes and 7 pairs of hooked legs. Females reach nearly 3 centimeters. Males are about half that. Every individual starts male. Juveniles enter a fish through the gills, and the first one to reach the mouth changes sex and becomes the female, a pattern called protandrous hermaphroditism. She settles at the base of the tongue and begins feeding on blood from the lingual artery. The tongue, cut off from its blood supply, atrophies until only a stub is left. What happens next is the part that gets this animal into every list of biological horrors, and it is genuinely true. The isopod grips the remaining stub and stays there. The fish uses it as a tongue. It presses food against the palate with the parasite's body, and it goes on feeding, and it goes on living. This is the only known case in the animal kingdom of a parasite functionally replacing a host organ. Confirmed hosts span at least 8 fish species, including the rose snapper of the Gulf of California. The animal became internationally famous in 2005 when one was found in a snapper bought in London, and it has stayed famous since, which is exactly why it belongs in a video about parasites people fear. So it is worth being direct about the danger, because the honest answer is narrower than the reputation. Cymothoa Exigua cannot infect a human being. It has never been documented inside one, it cannot survive in one, and cooking destroys it completely. There's no food safety pathway here at all. A legal dispute in the United States in 2012 over a specimen in restaurant fish was dismissed. The real risk falls on the people who handle fish for a living. The animal bites when picked up, and it bites hard enough to draw blood, and those wounds carry the ordinary marine infection risks, Vibrio and Mycobacterium species that live in seawater and enter through broken skin. Its relatives also cost aquaculture real money through suppressed growth and infested stock. Number 9. Nanophyetus Salmincola. On its own, this fluke is nearly beneath notice. It is under a millimeter long. In a human, it causes diarrhea, some abdominal discomfort, and a raised eosinophil count, and praziquantel removes it. It is also geographically trapped because its first host is a stream snail called Jugae placifera that lives only in a narrow band of the Pacific Northwest, and the parasite's entire world map is drawn by that snail's range. What makes it worth 4 minutes is what it carries. Inside the fluke lives a bacterium, Neorchezia helminthoishi, and that bacterium causes salmon poisoning disease. In dogs, it is close to a death sentence. Untreated mortality runs above 90%. Pacific Northwest veterinarians recognized the pattern in the 19th century, long before anyone could explain it. A dog eats raw salmon, and about a week later develops fever, discharge from the eyes and nose, vomiting, bloody diarrhea, and swollen lymph nodes. and then dies. Native communities in the region knew not to feed raw salmon to working dogs generations before that. The mechanism took until the 1950s to establish, when Philip and colleagues at the Rocky Mountain Laboratory identified the rickettsial agent. And the delivery system is the elegant, unsettling part. The bacterium does not have to find its own way into the dog. It is passed down through the fluke's own eggs transovarially from generation to generation, riding the parasite from snail to fish to mammal. The fluke is the vehicle, the bacterium is the passenger, and the passenger is what kills. Salmonids carry the encysted larvae in kidney, muscle, and fins. Definitive hosts include dogs, foxes, raccoons, bears, mink, and fish-eating birds. Human infection with the fluke was first documented in a 1974 report, and Eastburn, Fritchie, and Terhune published a human case series in 1987, traced to raw, undercooked, and home-smoked salmon and steelhead. Cold smoking never reaches a temperature that kills anything. And here's the question nobody has answered. Humans acquire the fluke, and the fluke is carrying the bacterium, and humans do not develop salmon poisoning disease. Something about human physiology stops it. What that something is remains genuinely unknown. Number 10. Trichobilharzia. This is the one you have probably already had. Swimmer's itch, properly cercarial dermatitis, is caused by the larvae of schistosome flukes whose real hosts are birds, mostly ducks and geese. Trichobilharzia regenti, sedati, and oceloti are the best studied. In the sea and in estuaries, the job is done by Ostrobilharzia and Ornithobilharzia, where the same condition is called clam digger's itch. Freshwater snails of the genera Limnaea, Radix, and Stagnicola release the larvae into warm or shallow water. Release is triggered by light and warmth, which means the water is most loaded in the late morning and early afternoon on the calmest, sunniest days. An onshore wind pushes the larvae into the shallows. That is precisely where small children stand for hours, and it is why outbreaks cluster at the shoreline rather than out in open water. The larva has a forked tail and it swims until it finds skin. Then it uses enzymes to digest its way through the outer layer, and it takes only minutes. Inside a duck, it would find a blood vessel and grow into an adult. Inside a person, it cannot. It gets a short distance into the skin, it stalls, and it dies there. The rash is not the parasite. The rash is your immune system responding to a dead animal in your skin. That distinction explains the pattern people find confusing. The first time you are exposed, you may notice almost nothing. The immune system has not met this material before. The second and third and 10th exposures are progressively worse because sensitization has occurred and the reaction escalates each time. Spots appear within hours, become intensely itchy papules over a day or two, peak at around 3 days, and clear within a fortnight. The main complication is a bacterial infection from scratching. That would be the whole story, except for one loose end. Trichobilharzia regenti is neurotropic in birds. It does not travel by blood. It travels through nervous tissue. Experimental work in mice has shown the larvae reaching the spinal cord in a mammal. Whether that happens in human beings is not settled, and it is not a comfortable open question for something this common. Number 11. The liver flukes. James McConnell opened the bile ducts of a Chinese carpenter in Calcutta in 1874 and found them full of flat, translucent, lancet-shaped worms. That was Clonorchus sinensis, the Chinese liver fluke. Its Southeast Asian counterpart, Opisthorchis viverini, dominates northeast Thailand, Laos, Cambodia, and central Vietnam. And a third species, Opisthorchis philippinensis, runs across Russia, Ukraine, Kazakhstan, and Siberia. Combined, they infect somewhere near 35 million people. The cycle is a freshwater one. Bithynia and Paraphoscereus snails release larvae, and fish of the carp family carry them in flesh and scales. In northeast Thailand, the dish is koi pla, raw minced fish mixed with herbs, lime, and spices. Eating it is not incidental to the culture, it is central to it. After a meal, the larvae exist in the duodenum and go looking for bile. They swim up the opening of the bile duct, the ampulla of Vater, and they are inside the liver's plumbing within hours. And then they stay. These flukes live in the biliary tree for 20 to 30 years, feeding on the duct lining, and for most of that time, the host feels nothing worth reporting. What is happening during those decades is slow. The ducts thicken, scar, and dilate. The lining cells proliferate, then become disordered, then become dysplastic. The International Agency for Research on Cancer classified Opisthorchis viverrini as a Group 1 human carcinogen in 1994 and Clonorchis sinensis in 2009. Group 1 is the definite category, the same tier as tobacco smoke and asbestos. The cancer is cholangiocarcinoma, malignancy of the bile duct itself, and Northeast Thailand has the highest recorded incidence anywhere on Earth. Some provinces exceed 80 cases per 100,000 people per year. The mechanism is argued three ways: physical irritation from the worms, chemical signaling from what they secrete, and nitrosamines generated by decades of inflammation. A granulin-like growth factor released by the fluke has been shown to drive host cell proliferation directly. Praziquantel kills every worm at a single dose. It does not unpick 30 years of scarring, and it does not stop people eating koi plar next month. The Lawa Model Project paired drugs with snail control, fish safety work, and education, and reported real reductions where the whole package was applied. Number 12. The broadfish tapeworm. This is the one people picture when they hear the word tapeworm, and the numbers earn the reputation. It exceeds 10 meters inside the human intestine, with older records running considerably longer. A single worm may carry more than 3,000 segments and shed up to 1 million eggs in a day. It was called Diphyllobothrium laudum for most of its scientific life, and was reclassified into the genus Bothriaceacephalus in 2017 on molecular grounds. It does not attach the way a beef or pork tapeworm does. There are no hooks and no ring of suckers. Instead, the head carries two long slit-like grooves called bothria, which grip the intestinal wall like a clip. From that anchor, the body streams downstream, absorbing everything it touches. The route in is a copepod first, then a freshwater or a migratory fish, and the classic sources are perch, pike, burbo, roofie, and salmon. Historically, this was a disease of the northern lake country, Finland, Sweden, the Baltic states, and northern Russia, and 19th century surveys in some lakeside communities found more than 1 person in 5 infected. Most of those people had no dramatic symptoms. Mild abdominal discomfort, some fatigue, occasionally the alarming experience of passing a length of segments. What makes this worm medically distinctive is a single nutrient. It competes directly for vitamin B12 in the intestinal lumen, and a large worm can absorb the overwhelming majority of what the host eats, in some measurements approaching all of it. The result is the only helminth infection that classically produces megaloblastic anemia. Red cells become large and ineffective, and because B12 also maintains the nervous system, severe cases develop tingling, unsteadiness, and in the worst instances, subacute combined degeneration of the spinal cord. Only a minority reach that point, around 2% in older Finnish series, and whether a given patient does appears to depend on genetics and on how high in the small intestine the worm has anchored. A single dose of praziquantel ends it. The interesting part is the direction of travel. Prevalence fell for a century as fish handling industrialized. Then sushi, gravlax, ceviche, and carpaccio went global, and case reports began appearing in Brazil, Chile, Peru, and across Western Europe in countries with no endemic cycle at all. Look at what all the 12 have in common. Not one of them reaches a human directly. Every single one requires at least 2 other hosts first, and in most cases those hosts are a small crustacean and then a fish. That is not coincidence. It is what water permits. Microscopic larvae die of drying on land within minutes. Suspended in water, they persist, drift, and are swallowed by filter feeders that concentrate them from enormous volumes. Copepods and snails serve as near universal first hosts because they are everywhere, and they eat everything small. From there, predation stacks the larvae upward. A fish eats thousands of copepods across a life, and every larva it acquires simply waits, unchanged in the muscle. Fish flesh is one of the best long-term storage media in biology. Then the hosts move. Migratory fish carry parasites across ocean basins. Birds distribute eggs across continents in a season. Whales and seals explain why acanthocephalans are waiting inside mackerel at all. The result is a set of cycles that were running long before people arrived and into which people insert themselves purely by eating. That accidental entry is why these infections are as bad as they are. In its proper host, a parasite arrives, settles, and stops. In a human, it frequently cannot find what it is looking for, so it keeps moving. And moving through tissue is what puts worms in eyes and spinal cords and brains. Being the wrong host is more dangerous than being the right one. The interruption points are few and well understood. Heat breaks every cycle here. Freezing breaks nearly all of them. Snail control breaks them a step earlier. What cannot be broken is the reservoir because birds, rats, cats, seals, and whales will carry these parasites regardless of what any human population does. There is a pattern running through the case literature that matters more than any single species. These parasites are dangerous out of proportion to how common they are because of what they are mistaken for. Paragonimus is treated as tuberculosis, sometimes for the better part of a year. Acanthocephalan infection is operated on as appendicitis. Angiostrongylus is admitted as bacterial or viral meningitis. Gnathostoma swellings are managed as allergy. Capillariasis is worked up as inflammatory bowel disease. Sparganosis is imaged as a tumor until someone notices the lesion has moved. Heterophyes eggs are read as clonorchus eggs down a microscope, and the liver flukes themselves announce nothing at all until a cancer arrives 30 years late. There is one shared clue, and it is easy to miss. Eosinophils, the white cells the immune system deploys against parasites, rise in the blood and, critically, in the spinal fluid. An eosinophil count in cerebrospinal fluid separates a worm from a bacterium quickly, but the count is only ordered if someone is already suspicious, and suspicion depends almost entirely on a dietary and travel history that is frequently never taken. That gap widens as cases travel. Imported infections turn up in hospitals where no clinician has seen the disease brought by travel, migration, and an international seafood trade that now moves raw product between continents in a day. Serological tests exist, but they live in reference laboratories. Polymerase chain reaction assays are improving unevenly. Delay is not neutral. In a migrating infection, every week of misdiagnosis is another week of tunneling, and treatment itself carries a trap, because killing a worm inside the central nervous system intensifies the inflammation, which is why steroid cover is now standard practice. The recurring failure in these cases is almost never the laboratory. It is the question nobody thought to ask. By this point, one question is doing all the work, so here's the practical answer. Heat kills every organism in this video. The working target is 63 degrees Celsius throughout the flesh. And throughout is the operative word, because thickness matters far more than time at the surface. Freezing is the accepted alternative, at minus 20 degrees Celsius for 7 days, or minus 35 degrees for 15 hours. Commercial sushi-grade fish is blast frozen precisely to meet that standard. Domestic freezers frequently fail to hold minus 20 reliably, which is why the home version of this is less protective than people assume. Everything else is unreliable. Salting kills some encysted larvae and misses others, which is what makes Fessick a recurring outbreak vehicle in Egypt. Cold smoking never reaches a lethal temperature at any stage, which is what put salmon flukes into Pacific Northwest patients. Marinating in citrus does not reliably kill anything in ceviche, and Gnathostomiasis in Latin America is the proof. Alcohol does not sterilize raw crab, and drunken crab dishes remain a route for lung flukes. Inspection helps in one direction only. Candling fillets over a light box catches large nematodes, and commercial processors do it routinely. It will never catch a metacercaria, because those are microscopic. Prompt gutting reduces the number of larvae that migrate from gut into muscle after a fish dies. The rough division is worth carrying. Freshwater and estuarine fish carry the higher liver fluke and tapeworm risk. Marine fish carry the higher nematode and spiny-headed worm risk. Snails, slugs, prawns, and crabs need thorough cooking with no exceptions, and leafy vegetables need washing because slug slime carries rat lungworm larvae onto salad that never went near an animal. The honest position on most of this is that the numbers are floors rather than ceilings. Why humans resist neorickettsia while dogs die of it is unexplained. Whether Trichobilharzia larvae can reach human nervous tissue as they demonstrably do in mice is unresolved. The true global burden of Gnathostomiasis outside active surveillance zones is guesswork, and deep-sea parasite diversity is almost entirely undescribed because most deepwater fish have never been examined for parasites at all, and whale and seal parasites are sampled overwhelmingly from animals that happen to strand on a beach. Meanwhile, the conditions are moving. Warming water extends snail and copepod ranges toward the poles, shortens larval development times, and lengthens transmission seasons. Aquaculture concentrates hosts at densities with no natural equivalent. Ballast water relocates intermediate hosts faster than any surveillance system tracks them. There is real disagreement in the literature, too. Whether cancer risk persists after liver flukes are cured is contested, with some researchers arguing that reinfection, rather than residual damage, drives the ongoing incidence. And at the speculative edge sits a genuinely inverted reading of the whole subject, that chronic low-level helminth infection was normal for the entire history of our species, and that removing it entirely helped produce the modern rise in autoimmune and allergic disease. That is the hygiene hypothesis. It has serious mainstream support, and controlled helminth therapy trials continue on the strength of it. The results so far are mixed and heavily disputed. Even the naming is unfinished. Molecular work keeps splitting species once thought single, and both Dibothriocephalus and the Capillaria genus placement remain live arguments. Nothing in this video is visible in the water. That is the part worth carrying away. The ocean's most dangerous residents, measured by what they actually do to human beings, are not the animals with teeth. They are small enough to be swallowed without being noticed, and every one of the 12 arrives the same way, through the mouth, in a meal. The vector is not the sea, it is the plate. None of them evolved for us. These cycles were built for whales, seals, rats, cats, dogs, and waterbirds, and they were running for millions of years before anyone was there to interrupt them. Human infection is an evolutionary accident almost every time, and accidents do more damage than adaptations do. A parasite that knows where it is going settles quietly and takes what it needs. A parasite that is lost keeps moving, and movement through tissue is what puts a worm in a spinal cord. What has changed is not the ocean. Refrigeration remains one of the most effective medical interventions ever deployed, and prevalence collapsed almost everywhere food chains industrialized. Then raw preparation became fashionable, and the trade in raw seafood went global, and case reports started arriving from countries with no endemic cycle whatsoever. The distance between the deep water and the kitchen has closed. What lives down there now travels further than it ever could on its own.